All right, welcome to the section on active transport. Now, we've talked about the movement of molecules, we've talked about diffusion, it's onward to active transport. Now, active transport uses energy to move solutes against their gradients. Now, facilitated diffusion is still passive because the solute moves down its concentration gradient. Even though it's being helped by transport proteins, we know that there's no energy being provided by the cell, no ATP. So that's still considered passive. Some transport proteins, however, can move solutes against their concentration gradient, from a low concentration to a high concentration. Now, when we move substances against their concentration gradient, we have to expend energy on this. All right? Just like a salmon swimming upstream, we are going against the current here, so we're going to need to expend energy to do so. Now, active transport is usually going to use energy from the cell in the form of ATP. Not always, but usually. Active transport is performed by specific proteins embedded in the membrane. These proteins are referred to as pumps, specifically pumps. So we know that if we hear if something is a pump, it's going to be active transport. The sodium-potassium pump is one type of active transport system, and it is one of the most widely used examples of active transport. Extremely important in the nervous system. All right, so this is the sodium-potassium pump. We need to recognize what's happening in the sodium-potassium pump to be able to understand active transport. So this pump is very efficient. It moves both sodium and potassium. Okay, so again, enzymes are substrate specific. Proteins embedded in the membrane are also going to be ion specific, molecule specific. So the sodium-potassium pump can only move sodium and potassium. Now, because it's a pump, it's going to move both of these molecules against their concentration gradient. Interestingly enough, it does not move both of them in the same direction. Okay, so let's take a look at number one. Cytoplasmic sodium binds to the sodium-potassium pump. So there are these specific binding sites. They are shaped like sodium. Sodium is going to bind to them. Now, until we get 3, nothing happens. Okay? Nothing happens. But when that third sodium attaches, we're off to the races. Okay? We're going to move. Now this is the cytoplasm. Sodium is being moved from the cytoplasm, which is where its low concentration is, out of the cell to where its high concentration is. We need to do this in order to allow the cell to function correctly. Now, when that third sodium binds to the binding site, ATP is going to be split, okay? And it's going to attach the phosphate group to the sodium-potassium pump, okay? This is a process known as phosphorylation, and we are going to be talking about phosphorylation a lot in the upcoming chapters. So get used to this. Anytime you attach a phosphate group to something, it's known as phosphorylation. You know that ATP contains energy. Well, along with the phosphate group being attached, we're going to give this protein some energy. Okay? So the phosphate group is the key in the ignition, and that's going to signal that we are activating this protein. And the energy to activate is coming from the ATP. Now, when I say activate, what are we doing? Well, you'll notice from step 2 to step 3, what happens? There is a change in the protein's conformation. Okay? There's a change in the protein's conformation. Similar to a carrier protein, we are changing the shape of the protein. Now, not only do we change the shape of the protein in terms of which way the opening faces, but if you'll notice, the binding sites change as well. They're actually going to squeeze, and in doing so, they're going to squeeze the sodium out. They're going to push it out, okay? So the sodium is going to exit the cell. You'll also notice that these binding sites over here have changed shape. They are now the correct configuration to pull in potassium ions, okay? So take a look at step 4. So this is where we're going to be pretty impressed by the efficiency of this molecule, okay? We're going to push 3 sodium out, And then we're going to bring 2 potassium in. So 2 potassium are going to come in. They're going to bind to the binding site. When the second potassium binds to the binding site, that's going to trigger the release of the phosphate group. Okay? Phosphorylation, attachment of the phosphate group is going to change the configuration, that can change the conformation to phase out. Releasing of the phosphate group allows the protein to return to its original conformation. Now, this is something important. We had to put energy in to change the protein's conformation to phase out. We don't have to use any energy for the protein to return back to its original conformation. It's snapping back, okay? It's actually releasing potential energy. The best way I like to use to explain this is if, as a kid, sometimes at a party you would get one of those little plastic things, and it was a popper, right? And you could invert the popper and put it on the table, and then after a little while, it would return to its original shape and it would pop, okay? You put the energy in to flip it, and then you just let it sit there until it returned back to its original shape and then popped. That's what we're doing here. We're paying for the change in one direction. The return to the original conformation does not require any further energy because it's just returning back to its original state. We actually added energy to make it into a less stable configuration, and then when it returns back to its original, more stable configuration, we don't have to pay for that. So what did we do? We got 2 moves for the price of 1. This is a steal, okay? So when we return back to our original conformation, okay, loss of the phosphate restores the protein's original conformation. That includes the binding sites. The binding sites are going to return back to their original conformation, which are going to squeeze the potassiums out, and they're going to come into the cell. When I say out, I mean out of the binding site, but into the cell. So the sodium-potassium pump pumps 3 sodium out and pumps 2 potassium in. Okay? Now, the mnemonic device for remembering this. I want you to write this down. Okay? Napkin, the word napkin, 32. Okay? Napkin, 32. This is how you're going to remember this. So, the first 2 letters, Na, that's sodium. Okay? The third letter, P, what happens to the sodium? It gets pumped out. Pumped out. P is for pumped out. Okay? Then the fourth letter is a K. That stands for potassium. Okay? What does potassium do? In, it comes in. Okay? And what are we working with? We're working with a 3-to-2 ratio. Okay? The 3 comes first because that's going to be the sodium. The 2 comes second because that's going to refer to the potassium. So sodium is pumped out. There's 3 of them. Potassium comes in. There's 2 of them. So this is going to allow you to answer any question that could be asked about this, right? Where is the high concentration of sodium? Well, if the sodium is being pumped out, the high concentration has to be outside because we're pumping, and pumping is active transport, right? Where is the low concentration of sodium? Well, sodium's coming in. The low concentration of sodium is outside. Which way does it go? Comes in. How many? 2, right? 3 sodium, 2 potassium. Napkin 32. That's going to be how you remember all of the movements of the sodium-potassium pump, okay? This is a very widely used example of active transport, and it's extremely essential for the nervous system. Okay, it's extremely important for the nervous system. So you would find this in neurons because you need to move the particles, the charged particles, in order to establish the charge of the electrical impulses, right? So we need to move the positively charged particles in order to reestablish polarization so we can send another neural charge. Okay, so recapping. Passive transport. We've got diffusion. Right? What moves by diffusion? This is simple diffusion, by the way. Simple diffusion through the membrane. CO2 does this. Right? It's a gas. It's small. CO2 diffuses. O2 diffuses. Small. It's a gas. No worries. Right? It's non-polar. Not an issue. And water. Water moves by simple diffusion. What about facilitated diffusion, right? Well, we could use water through an aquaporine. We could move glucose through a channel protein. Okay, this is a carrier protein. Both of these are facilitated diffusion using proteins to move molecules from their high concentration to their low concentration. And then we've got active transport. And what's our go-to example of the active transport? Sodium-potassium pump. Right? The sodium-potassium pump. Right? It's a pump because it's going to move things from a low concentration to a high concentration and requires the energy from ATP. Remember, there's no energy required for diffusion. We're just using the kinetic energy that's in the molecules already. But we need energy from ATP to drive active transport. All right, so maintenance of membrane potential by ion pumps. Membrane potential is the voltage difference across a membrane. We do have a significant amount of electrically charged particles. We've got sodium, we've got potassium, we've got chlorine, we've got calcium, and we've got hydrogen ions. So we're going to be able to move and use charged particles not only to create a concentration gradient, but also a change in charge, which is going to give us what's known as an electrochemical gradient. Okay? So two combined forces, collectively called the electrochemical gradient, drive the diffusion of ions across a membrane. The chemical force, which is simply how many ions or how many particles are on either side. That's the concentration gradient. And then the electrical force, the effect of the membrane's potential on the ion's movement. So we're going to get a differentiation in charge, and that's also going to get the particles to move. If it's going to move H plus ions, it's known as a proton pump. And again, I am laying the groundwork for our studies in the next section. When we get to the cell respiration and photosynthesis part of our studies, we're going to need to know this information. So not only are there more H pluses on the right than the left, but there's a greater charge on the right than the left. So that means the right is positively charged and the left, in comparison, is negatively charged. So an electrogenic pump is a transport protein that generates voltage across a membrane. The main electrogenic pump of plants, fungi, and bacteria is a proton pump. We are going to see the proton pump again. So we pay energy to move the H plus ions, and then we can use that H plus ion to help us do work or to move and create energy. So what can it help us with? So one example is co-transport. This is a coupled transport by a membrane protein. So co-transport occurs when an active transport of a solute indirectly drives transport of another solute. So we move one molecule and we pay for it, and then we use that energy to move another molecule. So plants commonly use the gradient of hydrogen ions generated by proton pumps to drive the active transport of nutrients into the cell. So let's take a look. I need to move sucrose. Sucrose is big. Sucrose is polar. Sucrose is not going to cross the membrane without some help. Okay? So I need a protein. I need a sucrose protein. But I need more than just that. The sucrose is big and not going to easily move. So what I can do is I can pump H plus ions into, in this particular case, outside of the cell, creating a high concentration of H plus ions. Now I let the H plus ions diffuse, and the diffusion of the H plus ions is actually going to pull the sucrose in. It's going to power the movement of the sucrose. So the sucrose H plus cotransporter uses the energy of the movement of H plus ions down their concentration gradient to move the sucrose in. So the sucrose is actually moving by facilitated diffusion, but we're paying for it with the movement of the H plus ions. So we need a cotransporter. Okay, we need a cotransporter. Is this active transport? Well, we're paying for it with ATP to make the high concentration of H plus ions, and then the sucrose goes along with that. So, ish. If you count the H plus ions, then yes. All right, we're almost done. Let's talk about bulk transport. Now, bulk transport across the plasma membrane occurs in one of two ways, or two ways actually. It's either exocytosis or endocytosis. Exocytosis is out of the cell, right? Exit, exocytosis out of the cell, the process of cell exiting. Endocytosis, the process of cell within or bringing in, okay? So, endocytosis is cell eating. Exocytosis, we are secreting things from the cell. So, small molecules and waters enter and leave the cell through the lipid bilayer by transport proteins, but if it's something big, right, large molecules such as polysaccharides and proteins cross the membrane via vesicles. So in exocytosis, transport vesicles migrate to the membrane, fuse with it, and release their contents. Many secretory cells use exocytosis to export their products. There are two main types of exocytosis, secretion and excretion. And it's extremely important that you know the difference between these two. Secretion is a product with a purpose. Excretion is waste. Okay? So think about humans. A secretion of humans might be tears, sweat, saliva, okay? Mucus. These are products with a purpose. They do a job. Excretion would be waste products, urine and feces. We're just getting rid of those. We don't make those to use them for anything. Okay? So that's the difference between secretion and excretion. Endocytosis is the cell takes in macromolecules by forming vesicles from the plasma membrane. Endocytosis is the reversal of exocytosis involving different proteins. So we've got three types of endocytosis. We've got phagocytosis, which is cellular eating. The cell engulfs particles into a vacuole. We've got penocytosis, which is cell drinking. Cell creates vesicles around the fluid. And then we've got receptor-mediated endocytosis, which allows for the binding of ligands to receptors that are actually going to trigger vesicle formation. Okay? In all cases, we're creating these vesicles. So here's a visual of the receptor-mediated endocytosis. How is it different? Well, in the other two, you're kind of just making a vesicle and you're swallowing whatever you can. You're taking in a bacteria. You're swallowing a gulp of fluids. Whatever that is, it's random, right? You're just making a vesicle and hoping for the best. In receptor-mediated endocytosis, you're specifically binding, okay, a particular molecule to this receptor. So in this particular case, I want the purple. I don't really need the green. Okay? If I get a couple green, that's fine, but I really want the purple. So when the purple has bound to all of the possible receptors, it signals the creation of this coated pit, okay, which is going to become a coated vesicle, and that's going to enable me to be more specific in the molecules that I am collecting. Okay, again, I might still get a couple green, but if I were to just randomly take a swallow and create a vesicle, I am not necessarily going to get the majority of the purple molecules, which is what I want. So receptor-mediated endocytosis is a very specific type of endocytosis that allows me to capture a particular molecule. Okay, so again, we've got phagocytosis, which is cell eating, like a cell eating a bacteria. We've got penocytosis, which is cell drinking, taking in fluids. And then we've got receptor-mediated endocytosis. This is the opposite of exocytosis. Okay? Bulk transport requires energy, but we're not using pumps. We're literally creating vesicles for eating and drinking and or sending things out, from secretory vesicles that are created from the Golgi. All right, take a look at that. If you have any questions, let me know.